So in this video, what we are going to talk about is the unfolded protein response for the secretory protein. So now, previously, we already talked about that how all the different secretory proteins are transported into the endoplasmic reticulum. It could be the soluble proteins, which are in the ER lumen in the aqueous solution, or it could be associated with the membrane. So here what we are talking about are the only proteins which are in soluble aqueous environment of the ER lumen. So that means that all of these proteins were transported with a signal peptide on, and that was cleaved, processed by the signal peptidase, and this protein is folded in the functional form to go to the Golgi. So we are talking about these proteins, that how they can actually quality control these proteins if they don't properly fold. So here, if we're just looking at the endoplasmic reticular membrane, so right here I'm just drawing just two phospholipids right here. So right here is your membrane, and you have your unfolded protein right here. Somehow it misfolded, it did not fold. So how the response of the ER will be to take care of this unfolded protein so that it is not accidentally transported into the Golgi. So here what happens, that there is an enzyme associated with the membrane. This is a membrane-bound enzyme. So they are right here onto the membrane, and these are inactive enzymes in the monomeric form, and we call this as IRE1. These are basically endonucleases, and they are in the monomeric form. And why they are in the monomeric form? Because there is a protein called BIP, binding protein, BIP. They are associated with these IRE1 monomers. So this is IRE1, this is in monomeric form. They are single. And of course, they are inactive. They are not doing anything in here. So now what happens, there are different types of these responses in the ER. It could be stress and other things as well, but here we are only talking about the unfolded protein response. So if this protein is unfolded, what happens is the BIP actually comes off from these IREs. So the BIP will not be there anymore. The reason being, this BIP will now bind to this, the BIP will bind to this right here, so to the unfolded protein, so the BIP. So the unfolded proteins by nature binds to the BIP, and it is dislodged from the IRE monomer. So this is BIP, this is BIP. This is now bound to the unfolded protein. So that actually helps these monomers to dimerize. So what will happen? This basically will find each other, and they will dimerize. They will dimerize. And this IRE1, this is the dimer, two of them. This is active. And what's the function for that? This is an endonuclease. This is an endonuclease. And so now it's functional, membrane-bound endonuclease, and its endonuclear activity is towards the cytosol, because this is your cytosol. This is the lumen of the ER. Now, there is one mRNA, and it's kind of a little bit unusual compared to the other, that is transported from the nucleus after synthesis in an unspliced form. What we mean by that? That this particular mRNA, which we called as the HAC mRNA, HAC or HAC mRNA, HAC1 mRNA, and this mRNA does have an exon right here, and it does have an intron right here. Intron. So this is in unspliced format. So what basically the endonuclease does, that it will cut right here and splice the intron out, and the exons will basically join. So now what you have is basically the spliced, and we also can mature HAC1 mRNA. So this is what we have. So what it's going to do now, so that HAC1 mRNA is going to basically translate it by the ribosome and going to make some transcription factors. Transcription factors. So this is basically the protein. Again, this is a protein, and this will be the DNA binding protein. This is synthesized in the cytoplasm, and basically this protein essentially is going to go back all the way back inside the nucleus. If I just draw, this is the nucleus in the cell, it will go back. That protein will be translocated by the nuclear transporter into the nucleus. Now what you have here, you have the DNA. It will bind to the transcriptional promoter region. It will bind right here, bind, and it will basically turn on some of the gene needed for the protein, unfolded protein response. So turn on genes required, and this is number of genes, just not only one. And some of them could be proteasome associated. Proteasome associated means that these are basically the machineries which will degrade the unfolded protein. Some of them are ubiquitin. I'm just not writing the whole thing. The ubiquitin ligase. So what happens, the ubiquitin ligase basically joins ubiquitin residues onto the protein. These are UBs. When a protein is basically bound with the ubiquitin, which is done by the ubiquitin ligase, they are marked for degradation. So they will be transported out of the ER, and the proteasomes, which are already synthesized by this entire response, they are going to degrade those protein. So that's your basically unfolded protein response in the ER.